Okay, so we previously discussed the capsule, and then the next layer under the capsule is the cell wall, which is a highly important component of the cell. So, as bacteria are single-celled organisms, unlike animal cells, they need extra protection, and the cell wall plays a vital role in this cell protection, protecting it from the harsh conditions outside. So, the bacterial wall is actually a target for antibiotics. Because humans don't have this cell wall, this means antibacterials can specifically target bacteria without affecting the human cells. So the key molecule in the bacterial cell wall is peptidoglycan, and this structure forms a mesh-like sac around the cell and provides it with rigidity. Peptidoglycan is a huge polymer, and it's made up of polysaccharides linked by peptide bridges. The polysaccharides are long sugar chains of alternating NAG and NAM units, which are firmly attached by chemical bonds. As shown in the diagram to the right, polysaccharides are arranged like cables that encircle the cell, and these are connected by peptide bridges made of 4 amino acids. Peptide bridges are conveniently linked to NAM sugars. So each individual unit of peptidoglycan is therefore a NAG-NAM tetrapeptide. Okay. The peptide bridge can actually be made up of different amino acids, and this actually helps create diversity between bacteria. So the peptidoglycan structure is actually different in gram-negative compared to gram-positive bacteria. So in gram-negative bacteria, the tetrapeptide chains extending from each NAM unit are actually directly cross-linked, whereas in the gram-positive bacteria, these tetrapeptide chains are linked by pentaglycine cross bridges. You see this in the diagram below. As we said before, humans don't actually have a cell wall, so therefore the cell wall is a common target of antibiotic treatment, and the antibiotics will usually damage or prevent synthesis of the cell wall. And what happens to the bacterial cell is then it becomes more susceptible to osmotic pressure, and then so water can come in and the membrane will rupture. So certain immune cells are able to detect the peptidoglycan, and then they can actually engulf and destroy the bacteria. However, with gram-negative bacteria, the cell wall is actually buried, so the immune cells cannot recognize this, but they can recognize the LPS contained in their outer membrane. We'll discuss that a bit later. So as well as having a slightly different structure of peptidoglycan to gram-negative cells, gram-positive cells also have a much thicker cell wall consisting of several layers of peptidoglycan. Gram-positive cells also have teichoic acids in their cell wall, and this provides them with increased rigidity as it stabilizes the cell wall. So as we said, gram-positive cell walls, structurally quite simple, contain thick layers of peptidoglycan with the teichoic acid embedded in there to give rigidity. The gram-negative cell walls, however, are structurally more complex as they contain 3 layers. So they contain an inner membrane, just a thin layer of peptidoglycan, but then they also have an outer membrane, and this contains lipopolysaccharides, or LPS. If you remember, we said previously that the immune cells in humans can't recognize the peptidoglycan in the gram-negative cells because this peptidoglycan cell wall is buried. Instead, the immune cells recognize the LPS. Okay, so mycobacteria have a special feature. they actually have an additional layer of waxy mycolic acids in their cell wall. And this actually makes them quite difficult to stain, as the gram stain can't penetrate the waxy cell wall. So they require different staining. They require acid-fast stains, which then can penetrate the mycolic acid. You can see the staining of the acid-fast bacteria in the diagram below. And as well as being difficult to stain, mycobacteria are also difficult to treat, and they don't really respond to conventional antibiotics, and instead you need a specialised antimicrobial drugs or antimicrobial regimens to kill those. One mycobacteria you might have heard of is TB, tuberculosis, so that's a type of mycobacteria. This is less of a problem in the UK. There are more cases in developing countries, but then nevertheless there are some cases here. And you may come across these as a pharmacist. Okay, so we'll next discuss the plasma membrane of bacteria. So the plasma membrane of bacteria is actually similar to eukaryotic plasma membranes. It is a selectively permeable membrane, so it controls entry to the cell. So it acts as like a gatekeeper, protecting the cell from its surroundings and keeping important molecules from leaving the cells. So the structure of the membrane is actually described as a fluid mosaic module. And this is because the membrane components can move fluidly within the plane of the membrane, and also because of the mosaic-like components, which include several different lipids, proteins, and carbohydrates. So the plasma membrane consists of a phospholipid bilayer and some associated proteins. And you may have done a bit about this at A level, and we'll cover phospholipids in more detail in your lipid lectures. And approximately half of the membrane is composed of proteins, and these include recognition proteins, enzymes, receptors, carriers, and channels. This slide shows a good diagram of the plasma membrane, and you can see here the phospholipid bilayer, and you can see the array of different lipid, protein, and the carbohydrate chains. Okay, so we talked about this membrane being selectively permeable. So how do molecules cross the membrane? So there are three main methods by which they can cross. So there's simple diffusion, facilitated diffusion, or active transport. So simple diffusion is when molecules pass through the membrane down their concentration gradient. So we're talking about from a high concentration to a low concentration. And this process doesn't actually require energy and doesn't require a protein channel or a pump. So here we're talking about small uncharged molecules that can just pass easily through the phospholipid layer, and lipophilic molecules also can pass easily through the membrane in this way. However, if a molecule is charged or large, then they won't be able to cross the phospholipid bilayer easily. So they will require some kind of help. So either a selective protein channel or carrier. And these carriers will shield the molecule from the hydrophobic part of the membrane and allow them to cross. So this is called facilitated diffusion. Okay, facilitated diffusion. But it's important to know that even though they're using these carriers or channels, the molecules are still following their concentration gradient from high to low. So this process doesn't actually involve energy. Okay, so it's not an active process. And then lastly, you've got the active transport method, where molecules go across the membrane against their concentration gradient, so from low to high concentration. And unlike these previous two processes, so unlike the diffusion and facilitated diffusion, active transport does require energy. and does require protein pumps to pump the molecules across the membrane. And you can see these pumps in the diagram below. Okay, so next we'll talk about the nucleoid in more detail. So this is the region of the cell that contains the bacterial chromosome. So bacteria have a single chromosome, which isn't bound by a nuclear membrane, as we said earlier. And the nucleoid-associated proteins are the most abundant DNA-binding proteins, and they bind with bacterial DNA to organize the chromosome. And they do this in response to a change in environmental conditions. So, as we said before, bacteria may also contain extra chromosomal DNA. Okay, so that's DNA in addition to the main genome. And this extra chromosomal DNA is present as plasmids, which is small circular double-stranded DNA. There are often hundreds of these in the cell, and these are separate to the main genome. So when a bacterium divides, the daughter cells receive a copy of each plasmid. And bacteria can also transfer these plasmids one to another in a process called conjugation as well. So in this way, the DNA can be passed on. So you may have actually heard of plasmids in relation to recombinant technology, recombinant DNA technology. And so this is where scientists have actually taken advantage of plasmids. Okay, and they've used them as tools to clone, transfer, and manipulate genes. So the way in which they do this is researchers can actually insert genes into a plasmid vector, as it's called, and this creates what's called a recombinant plasmid. And this plasmid then can be put into the bacterium. And then when the bacteria divides rapidly, this then copies the gene in large quantities. So an example of this that's relevant to pharmaceutical drugs is the production of an insulin for diabetes. Okay, so this is how they now produce insulin, rather than using animal insulins, which what they used to do. So next we'll talk a little bit about bacterial ribosomes. So ribosomes are responsible for protein synthesis. Bacterial ribosomes actually differ in structure and size from eukaryotic ones, which, similar to the cell wall, this makes them a target for antibacterial therapy. So some antibiotics bind to ribosomes and inhibit protein synthesis, and in this way they inhibit bacterial growth. Okay, so there are a few antibiotics in this category. So in this way, the antibiotics can specifically target the bacterial ribosome to inhibit protein synthesis without actually affecting protein synthesis in the human cells. So some prokaryotic cells, mainly bacteria, have the ability to store excess nutrients in structures called inclusions, which are present in the cytoplasm. And these are important because they allow bacteria to survive in unstable environments. So lots of different examples of these. So I'll just mention a couple. So one is sulfur granules, and these store elemental sulfur. Can be used for metabolism. And then another example is gas vacuoles, which allow the accumulation of gas, and this actually gives bacteria more buoyancy in water. So this allows them to survive in water. And as I said, there are lots of other examples, which you can read through yourself. And here you can see images of these inclusions under a microscope. So bacteria are generally observed as vegetative cells. And what we mean by vegetative is that the cells are in a state to grow and reproduce. But certain bacteria do have the ability to form spores when they encounter unfavourable conditions. And these are the structures which protect the bacterial genome in a dormant state. And these can actually last for thousands of years. The spores don't need food or water, as they are not metabolically active, and they cannot grow and reproduce. However, these spores are very difficult to destroy and withstand very high temperatures, chemicals, and radiation. And the process of bacteria becoming spores is called sporulation. And the first step in this process is the formation of the septum, and this divides the cell asymmetrically, and this separates the DNA forespore. Forespore is the bit that's going to become the actual spore. So this is separated from the mother cell. A second membrane then forms around the forespore, followed by a protective cortex. Another coat then forms around the cortex, so this is a protein coat. So you can see really how well the endospore is protected and why it's actually really hard to actually destroy the spores. Once it's received this final coat, the endospore is then released from the mother cell. On this slide, we've got a brief summary of the sporulation process. Okay, so it's really important that you know the steps of this process. Okay, make sure you know that. And we've also got a close-up image of a spore on the left. And we've also got an image of a vegetative cell producing endospores. Right. The endospores are the small circles in green. So as we said, the endospores can survive for many, many years. And when living conditions become more favourable, they can undergo germination. And this is when the spore breaks hibernation and becomes metabolically active again. So it is now a vegetative cell again and can grow and divide normally again. And obviously at this point it does lose its resistance to the environmental stress that it had previously. So there are several clinically significant endospores. And one of these important ones is Clostridium difficile, and this has been a big problem in hospitals in the UK. So we call this C. diff for short, and this can be present in the normal intestine. But what happens is overgrowth of the C. diff can occur in a person's intestine, either because they're immunocompromised, or it could be because they've had a long course of broad spectrum antibiotics, and that's knocked out all their good bacteria, so to speak, so has disrupted the normal bacterial flora. And overgrowth of the C. diff can then cause symptoms such as diarrhea and inflammation of the bowel. And in severe cases, this can cause perforation of the bowel and even death, so it can be very serious. So when these bacteria pass out of the body, so pass through the intestine and pass out, they actually form spores. And like we said for spores, these can survive on surfaces for a long, long time. So they can survive in surfaces in hospitals, in bathrooms, on wards, and it's very easy to transfer these spores from one person to another. Okay, and then if a person ingests these spores, they can then become very ill. So these types of pathogens are very difficult to combat because their spores are so hard to deal with, basically. It's very hard to destroy them.